This is just the last little bit of recombinant DNA tech, but this is the exam questions to go with it because I missed this off the end of the other video. Okay, so just a quick look at these questions. I'm going to be popping in and out of them because they're quite long questions that cover lots of other genetic technology stuff that we haven't covered yet. So we'll probably return and revisit some of these at a later date, but we're just going to look at the recombinant DNA tech questions based on this. So this first question, some population of flies are becoming resistant to insecticides that intend to kill them. And what scientists are doing, they're developing a method for finding out whether a fly was carrying a recessive allele that gives resistance to an insecticide or a dominant allele that does not give resistance. So scientists cross the flies with the dominant alleles and the recessive alleles. They obtain DNA samples from parents and offspring, and they use the same restriction endonuclease enzymes on each sample to obtain DNA fragments. Now, the first question on this is, explain why the scientists use the same restriction endonuclease enzyme on each DNA sample. So that's 2 marks. And the reason that they've done this is if we think restriction endonucleases go in and they cut that DNA, they cut it out, they cut that required sequence out. So if we use the same restriction endonuclease enzymes on each DNA sample, that makes sure that we are cutting the DNA at the same base sequence. So therefore, all the fragments that we get will have the required gene on them. So I'll highlight where we get the 2 marks in this question. The first mark is for saying we're cutting the DNA at the same base sequence by using the same restriction endonuclease. And the second mark is saying, therefore, we will get fragments that all have the same required gene in them. And that will be the 2 marks for those. We'll skip past the rest. Actually, no, we won't. We'll look at this bit as well. So the scientists added 2 different primers to each sample of DNA fragments for the polymerase chain reaction. The same primer A3 only binds to 195 base pair fragment, and primer A4 only binds to 155 base pair fragment. The scientists separate the DNA fragments produced by the PCR on a gel, where shorter fragments move further in given time, and the results are here. And it asks us, explain why primer A3 and primer A4 only bind to specific DNA fragments. And we will cover this in a bit more detail again, but I want to quickly do this one as well. And the answer here is that each primer has a specific base sequence. So that will give you your first mark. So each primer has a specific base sequence, and that base sequence is complementary to the allele. Therefore, we only bind to those specific DNA fragments, giving you the full 2 marks on that question. We'll skip past the rest of that because it's looking more like PCR and lots of other different information that we don't need to do at the minute, but we will cover it at a later date. That then takes us on to question 2. Question 2 is looking at silkworms, and they secrete fibres which are harvested to manufacture silk fabric. And scientists produce GM silkworms that contain a gene from a spider, and the GM silkworms secrete fibres made of spider web protein, which is stronger than normal silk fibre protein. The method the scientists use is shown in the figure below. So they isolate the gene from a spider, insert the spider gene into a plasmid with a marker gene called EGFP. This gene codes for a protein that glows brightly in ultraviolet light. Inject copies of this plasmid into many eggs of silkworm, and then allow silkworm eggs to take up the plasmid to grow and produce spider silk. So the first question is, suggest why the plasmids were injected into the eggs of the silkworms rather than into the silkworms. So that is the part, this part here about step 3. Insert copies of the plasmids here. Now, the reason that we inject the plasmids into the eggs and rather than into the silkworms themselves is if we inject the plasmids into the eggs, that gene will then be present in all the cells or most of the cells of that then growing once that egg is fertilized, and then we get a new silkworm. We will then have the genes for that silk, the spider silk, being present in all or most of the cells of that organism. So that's our first mark. And the important part of that is so that it goes into the cells that make sense. And what we mean by that is going to cells that make sense is going into the cells that we would require it for. So the ones that make silk probably makes more sense than me saying make sense, that make silk. We don't want it going into ones that are just go into maybe like heart cells, for example, because that's not useful. So we want it to go to a specific place. And then it asks us to suggest why the scientists used a marker gene and why they use the EGFP gene. Well, as we know, when we're doing this plasmid work with recombinant DNA, not all of those eggs will successfully take up the plasmid. That's your first mark. So we use a marker gene to let us know that the silkworms, to identify the silkworms that have taken up the plasmid. And they use the EGFP gene because that's a phosphorescence and they will glow. So the silkworms that have taken up the plasmid will glow, and that'll get you 2 marks. So the scientists ensure the spider gene was expressed only in cells within the silk. What will the scientists have inserted into the plasmid, along with the spider gene, to ensure the spider gene was only expressed in the silk glands of the silkworms? And that, as you know, is a promoter region. That's just 1 mark for that. And then you're asked to suggest 2 reasons why it's important that the spider gene was expressed only in the silk glands of the silkworm. First reason is so we can harvest this protein really easily. If it's been expressed somewhere else in the silkworm, it's really not useful and we're not going to be able to harvest it easily. The second reason is those silk fibers, those spider gene silk fibers, if they were present in other cells, they may cause harm to the silkworm and cause the silkworm to die. So that would get you that final 2 marks for that question. The last little bit we'll do is just quick part here. So scientists want to measure how much mRNA was transcribed from an allele. Scientists isolate mRNA from the cell. They add an enzyme to mRNA to produce cDNA. Name the type of enzyme used to produce the cDNA, and that is reverse transcriptase. So that was just, I think, a really quick overview of these questions. There's just one more that I want to have a quick look at. So that's haemophilia. We use factor IX as a protein to treat haemophilia. And we'll look back at this question in a second. So they want you to name the type of enzyme that is used to cut the gene for factor IX from human DNA. If we have a look here, stage 1, gene for factor IX is cut from human DNA. Well, we know that we use restriction endonucleases in that situation, so that get you 1 mark. And then in stage 2, a jellyfish gene is attached to the human gene. So the jellyfish gene attached to the human gene codes for a protein that glows under fluorescent light. Explain the purpose. Same as that question before. The purpose of this gene is to act as a marker gene to show that the desired gene has been taken up. Only embryos with the gene will fluoresce, so then we know which one's which, and that is the 2 marks that you'll get for that question. And we will leave that there. All the rest are a bit more specific to other things, so we'll cover those at a later date.